Welcome. We present DREAM, a novel method for visual decoding of brain activity, which relies on reversing the human visual system. A machine can record the brain activity of someone caused by visual stimuli. Here, a picture of giraffes. Visual decoding consists into reconstructing the viewed image using only the brain responses, usually encoded as voxel. Solving such tasks would not only enhances our comprehension of the human visual system, but also establishes links between human vision and computer vision. Still, the small fMRI dataset and the intricate nature of brain activity still represent significant challenges to obtain fine-grained and accurate visual decoding. Recently, using pre-trained diffusion models led to significant improvement, as it offers better high-level semantics, meaning a coarse description of the image content. Still, the images reconstructed by such method, here the recent brain diffuser, suffer from important color and spatial inconsistencies when compared to the test image viewed by the patient. On the leftmost example, notice how the reconstruction accurately depicts a cow herd next to a forest, but fails at inferring the correct position and skin color of both the animals and surrounding vegetation. The same happens in all the other examples. To mitigate the loss of color and spatial information, our method, coined DREAM, addresses visual decoding using principles of the human visual system. Specifically, we introduce a novel formulation based on the fundamental knowledge of human perception, which seeks to reverse the pathway specialized in extracting semantics, depth, and color cues directly from the fMRI data, thus mirroring the forward process within the human visual system. As a result, DREAM outperforms state-of-the-art in visual decoding. Here, we present a schematic view of the forward pathways in the human visual system. When humans perceive the visual world, the retina within the eye captures incoming photons and converts them into electrical and chemical signals, which are then relayed to the brain, resulting in visual perception. Schematically, color and depth information are processed through the parvocellular and magnocellular pathways. The deciphered information then channels into the visual association cortex, VAC, for retrieving high-level semantic content from the visual stimuli. In our method, depicted on the right, we seek to inverse processes within the human visual system, thus deciphering concurrently the semantics, color, and depth cues from the fMRI recordings. Considering fMRI data as input, DREAM uses three components. The reverse VAC extracts semantics in the form of a clip embedding. The reverse PKM deciphers color and depth in the form of coarse spatial palette and depth maps. Finally, the reconstructed images is obtained by feeding the deciphered cue into a T2I adapter in conjunction with stable diffusion. We now provide a walkthrough of each component. The role of our reverse VAC component is analog to that of the VAC in the human visual system. In other words, it seeks to extract semantics from the fMRI data. This is achieved by training an encoder to align fMRI features with the pre-aligned and fixed clip space using contrastive learning. The initial contrastive loss aligns the fMRI embedding with both the clip image embedding and the clip text embedding. However, different from the literature, we address the problem of imbalance of data between the original clip trained on 400 million samples and standard fMRI dataset having only a few hundred samples. To combat imbalance, we employ a mixed code data augmentation strategy, which generates mixed fMRI data from convex combination of two fMRI data samples. While this allows extracting semantics, such knowledge is inherently bounded by the clip space capacity, unable to encode spatial color and geometry. Hence, we propose the reverse PKM component, which goal is specifically to invert the parvo, conio, and magnocellular pathways in the human visual system, thereby enabling the prediction of color and depth from fMRI data. We approach color and depth prediction as an RGB estimation problem. To combat the data scarcity issue of the fMRI modality, we introduce a multi-stage encoder-decoder training. Given limited pairs of fMRI and images with depth map, in the first stage, we train an encoder to map RGBD to the corresponding fMRI data. In the second stage, we train a decoder to reconstruct RGBD from fMRI. Finally, in the third stage, we fine-tune the decoder using external natural images in a fully self-supervised way. Altogether, the reverse PKM learns the fMRI to RGBD mapping, thus deciphering color and depth cue from fMRI. Given the deciphered semantics, depth, and color cues, we formulate reconstruction as a conditioned image generation problem. We leverage here the powerful stable diffusion using a pre-trained T2I adapter to inject the semantics, depth, and color cues as guidance of the generative process. The resulting image is the output of DREAM. To evaluate the performance of our method, we show the reconstruction result of DREAM in comparison to the most recent baseline. For qualitative comparison, we show also on the left the test image seen by the patient. DREAM can accurately decode the visual stimuli from fMRI much better than any other baseline, with better spatial and color arrangement, but also better semantics. For example, note how the reconstruction of the dish image is more accurate with DREAM and the only one that preserved the spoon. On the right, DREAM's reconstruction of the baseball field is the only one to accurately show only 3 players. Same performance boost is observed quantitatively, showing DREAM performs better on high-level metrics and on par or better with low-level ones. In the bottom 2 rows, we note that each of our components contribute to the best performance. We report detailed qualitative results for 2 examples, also showing variations of the reconstructions. The depth and color prediction from the reverse PKM are aligned with the pseudo-ground truth, despite the extreme complexity of the task. As a result, all DREAM reconstruction closely match the test images. For comparison, we deactivate the color guidance in the last 3 columns, which highlights its benefit. Note that faithfully recovering the original depth from fMRI is nearly impossible due to the information loss in capturing the brain activity. Still, we note that our coarse depth is sufficient to guide scene structure and object position, such as guiding the giraffe location and orientation. Likewise, the coarse color palettes provide a reliable constraint and guidance on the overall scene appearance. In the table below, we quantify the benefit of using our predicted semantic depth and color, which aligns with our qualitative judgment. Here are a few additional results of DREAM decoding. Notice how our method produces consistent results in terms of semantics, appearance, and structure. To evaluate the spatial and color quality of our reconstructions, we further compare the color and depth when predicted either from the decoded images or from the unseen test image. Our method DREAM consistently outperforms the very recent brain diffuser and mind-eye baseline. Please refer to our paper for more evaluation and ablation studies. We have also made our code publicly available. Thank you for watching.